I think we should start. 好，那大家中国的听众晚上好，我们美国东海岸的听众早上好，非常感谢百思会给我这个机会来跟听众们一起沟通和交流我们在抗击新冠病毒上的一些学习和知识的一些体会，因为这个演讲呢是我们是中美同时进行的，那为了更方便于交流，我这个演讲会用英文。如果我们中国的听众希望再听得更仔细一点，那讲完以后我们会把它回放成中文的这个字幕，所以呢会给观众有另外一个机会。所以呢在下面呢我就开始用英文演讲。好，谢谢大家。Okay, let's begin. Today, I really greatly appreciate the opportunity to have this discussion with our audience, especially in both US and China. It's a special situation where we face the same elements, which is coronavirus. And then we definitely need to share the learning from each other and then work together against these common elements. Today, I will be focusing on sharing the learnings and knowledge about GM-CSF antibodies for the treatment of cytokine release syndrome associated with COVID-19. Before I start, maybe just a few seconds about myself. I'm Zhong Shen. I'm the current CEO at l-Mab. l-Mab Biopharma is a company focusing on the innovative drug development, focusing in the area of immunologies, which differentiated itself from others by having the focus of immuno-oncology and autoimmune diseases with the best-in-class or first-in-class potentials. So we are young companies, and we have our self-developed discovery and development. So today I will be sharing with our learnings about our own innovative compound for the development of treating the coronavirus associated CRS. Today my discussion will be focused on a few aspects. First is the challenges of the patient treatment in severe COVID-19. And secondly, it will be the relationship between the cytokine storm and the severe COVID-19. And thirdly, the cytokine storm and the GM-CSF and its antibody as a potential treatment. Fourth is our experiences, how these GM-CSF antibodies being developed for the indication. Especially, we'll walk the audience through the concept, the strategies, and the development of protocols and the interaction with the regulatories, and which come to where we are now. I'm sure we are all aware the severity and the impact of COVID-19 in the patients. So since this January, we've been tracking this outbreak in China first. And as we collect more information, it is clear among these patient pool, 10 to 20% of them will develop severe or critical conditions, which requires intensive care. And many of them cannot be treated adequately, resulting in the mortality rates about from 2 to 10%. So if we look at the data so far available, we'll see among those critically ill patients, there are a lot of data showing that 80% of those patients actually have a profound lymphopenia, which is definitely has correlations with the immunodysregulations. And then also there are like cytokine storm, which has been spread out through all the cytokines. But of course, among them, GM-CSF and IL-6 stand out as the most significant. And we've also seen the from the pathogenic studies and then a lot of granulocyte infiltrations and also GM-CSF dysregulations, which has been reported by a lot of Chinese scientists at the front line of the outbreak, especially from Wuhan. So among all the cytokines, we have observed that GM-CSF is a driver which results in the inflammations and also resulting in this multiple organ failures, including lung, liver, and kidney, and heart disease. And I'm sure you have heard those incidences, patients died all of a sudden because of those conditions. And so far, there's no efficacious treatment. And recently, there are some suggestions about IL-6 and GM-CSF antibodies treatment for that, and I'll share with you later. But at the time when we had this outbreak in China, nobody had any data yet. But from the literature, we knew that GM-CSF has the potential to treat cytokine storm initiated by the CAR-T treatment. So we believe there are so much similarities among these two cytokine storm. There's potential we should maybe utilizing this for the treatment. That's how the project is initiated. Then we continue to collect information from the frontline and of those patients and the research. And we had particularly collected information from 3 studies. Those are all from Wuhan, from different research institutes. And if you look at these tables, which should be available to you, and you can see the first studies, they collected the information as of January 29th in Wuhan. Among 1,099 patients, 173 are considered as severe cases. And then from another studies, overall, there are a patient pool of 138, and then another one's 99. And then you can tell the severity or requires ICU treatment are about 15, 26, and 33%, which is pretty consistent from other reports afterwards. So we are looking at about average of 20% of patients who have been deteriorated from the conditions. So it is very critical that we intervene in the earlier to start to prevent it from becoming more severe, which could be too late to treat afterwards. So if we go to the next slide, we've seen that in China, we have been updating the treatment guidelines very quickly. And at the latest version, which is version 7, the treatment guidelines clearly introduced immunotherapy among all the other treatments. So introducing this meaning that immunotherapy treating these severe patients is very important. But at the time, all those immunotherapies are in the experimental period time. And if we move across the ocean, look at the US side, and as the outbreak spread out to the rest of the world, and then the US major cities are also suffering from these pandemics. And then quite a few guidance have also been coming out. Among one of those is from the US Center for Disease Control and Prevention. As you can tell, this guidance is very, very similar to what we observed from China. They also suggested incubation time 14 days, and the presentation of the disease has been categorized as common, uncommon, and unknown. And we all know the most common ones are like fever and some of the other, the SARS, and then like diarrhea. And then also the ill severity is categorized to mild to moderate, which accounts for about 80%. And then the rest of them are either severe and critical, which altogether accounts for about 20%. As we speak to the frontline physicians and the KOLs, they clearly told us. So it is, once the patients come into the severe critical stage, it's very hard to reverse. So they are desperately looking at the therapies which can prevent from deteriorating or detect when the patient could be deteriorated. So that's why we need to find something to intervene as early as possible to differentiate patients who can be potentially become severe. So as we can tell, once the patient becomes severe, the deterioration is very, very fast. And then we also need to realize the risk factors for this severe illness for the patients who are in age, cardiovascular disease, diabetes, chronic respiratory disease, hypertension, etc. are all those risk factors for this disease to become severe. And in US, there's multiple guidance and clinical management, usually being customized by the hospital individually. But overall, there's an infectious disease Society of American have the clinical management guidance people followed. So far, for the severe disease, there's no FDA approved drugs so far. And what we provided was the supporting care, and also based on the IDSA or WHO guidance. For those admitted patients to the hospital, currently we all know under the urgent approval by FDA, it is allowed to use hydrochloroquine in the context of clinical trial. However, they are recently complaining about all the potential risk about those very severe adverse events, especially for the cardiovascular and adverse events. So it is not ideal to use this unless there's some, we ran out of options. So I wouldn't go to details of all of those medications under the clinical trial or under so-called compassionate use conditions. But I think recently there are quite a few clinical trials come to place, which is IL-6, tocilizumab, and also the GM-CSF treatment. So for l-Mab, our GM-CSF antibody TJ003234 was approved for clinical trials in the middle of March. So it is just about to be initiated. Speak about GM-CSF. Let me just walk you through some of the key mechanisms of actions. So to help us to understand why we chose this, what is the difference between GM-CSF and IL-6. So if you look at the cytokine release cascade, GM-CSF is standing on the top of it, on higher hierarchies to control the overall cascade of the cytokine release. So if you, for IL-6, however, it is just one of those cytokines on the way. So we believe by these scientific theories that GM-CSF can have a better control to control the release of this cytokine, just like the switch on the higher levels. And then the next slides, if you recently, actually the presentations have shown they collected patient data from the COVID-19, and then they show the multiple cytokine release panels. GM-CSF is one of those have significant increase of those patients. So this is one of those demonstrating that GM-CSF played a very significant role. So if you look at the size 11, you comparing what has been dysregulated in the patient and what has been suppressed by introducing the treatment, you can tell the GM-CSF did play a role to suppress or control the cytokine release. And it demonstrates there's a potential to treat the patients who have been deteriorating in these COVID-19 situations. So in February, one of the research led by Dr. Wang Fushen in China showed that histological changes in those patients who have died of those severe COVID-19 And then you can tell from the slides here, there's bilateral diffuse alveolar damage with the cellular fibromyxate exudates, which is symbolizing a very severe inflammations. Underneath are the cytokine panel. You can tell there was a significant increase of the high inflammations and with the increase of the cytokines. So those two gave much more direct evidence that the hyperinflammations have been play a very critical role in the patients in the deteriorations, and also the cytokine playing a role in the correlation. So since then, there are quite a few clinical trials have been initiated with IL-6 or GM-CSF. One of those trials, which was led by Roche, with its tocilizumab, it is shown that actually patients have been improved by introducing this treatment, even though it's currently still preliminary with limited data. However, there are larger studies has been initiated based on that. So coming back to the GM-CSF, we knew it was originally developed for multiple autoimmune diseases, and recently has been introduced for the potential cytokine syndrome, release syndromes associated by the CAR-T treatment. So this mechanism is also being investigated under that situations. So if you look at this, graph, you can see cytokine release by GM-CSF has been, in multiple pathways. And then the first one, by introducing GM-CSF antibodies, you actually can suppress the activation and then proliferation of the macrophage in inflammatory tissues associated with this cytokine. And secondly, it can actually inhibit the myeloid cell differentiation. And then thirdly, is when there's migration of those inflammatory cells, which result in a large accumulation in the inflammatory tissues, GM-CSF neutralized antibodies actually can suppress that to further reduce the inflammations. And this one was from the Humanigen when they started to collaborate with Kite for the cytokine release syndromes initiated by the CAR-T treatment. It is obvious their preliminary research also showed that GM-CSF played a very significant role in the inflammatory cascade. As a result, it initiated a phase 1 study to evaluate its role directly and also on the COVID-19 patients right now, but started earlier last year's. They actually is doing that in the combination with CD19 CAR-T treatment in lymphoma patients, which is currently ongoing, I believe. And so we had a comparison with the GM-CSF antibody and the L-6 antibodies. So actually in our pipeline, we also have L-6. By comparison, we decided the GM-CSF is a better candidate for treating the cytokine release syndromes, as the reason I had already explained earlier. So one of the major key thing is for IL-6, they have no effect on the macrophage activation initiated syndrome. However, for GM-CSF antibody, it can do that. So it gives another layer of the strength to suppress the cascade of the cytokine release. Yet, it does not have the side effect as we've seen in the dexamethasone. So that will give us a better precise inhibition of the cytokine. So based on all the information we have obtained, we knew that GM-CSF needs to be initiated for this treatment is the in this COVID-19 patients. Actually, after we initiated the project, we have just to keep receiving this new informations. And then one of those most recent information we have obtained is actually a direct demonstration that GM-CSF worked for the patients. So this is a study conducted in Italy. It's an investigator-initiated study. Among the 6 patients treated with GM-CSF, they call them maralizumab, that all the patients show an early resolution of fever and the improvement in oxygenation within 1 to 3 days. None of those patients have progressed to require mechanical ventilation. That is so critical to keep the patient from deteriorating. So as a result, they believe this GM-CSF have a direct effect on like treating the patients with severe disease and also preventing the patient from further deteriorating. So actually, as a result, Kineska Pharmaceuticals, who is the sponsor for this study, their stock price went up to like more than 30% in a single day because of the release of the news. So we were pleased that the patient data supported our initiations of the study. So currently, there are multiple studies targeting on cytokine release syndromes correlated with COVID-19. And if you look at the table on the right side, it showed all the current studies and the sponsors in these areas. Altogether, I think the most common ones is IL-6, and the next one is GM-CSF. IL-6 because it's also a approved drug, so it has accumulated a lot of more safety data. That is why it become much easier for the sponsors and the regulatory agents to recognize it and then to approve these clinical trials. For GM-CSF, so far none of those has been approved. That's why it takes longer time to actually maximize its potential. So even for us, we had multiple interactions with KOLs, with the regulatory agents, trying to understand the safety profile, and which I will share with you later. So now there's compound which is developed by our company about 4 years ago. This is a GM-CSF antibody starting from our lab. Last year, we went to US for the first in-human studies. It is completed and demonstrated a very straightforward safety profile. And in those ranges, we had conducted a study, which is 1 to 10 milligrams per kilo in healthy volunteers, including a Chinese subject. So the overall study showed that it is well tolerated in those range studied, and also the PK/PD profile is linear and correlated very well. So it is supported that our study next for treating the patients with severe COVID-19, because first, we are not introducing any higher dose than the study that we had done the study before. Secondly, it is only single dose injections, so we're not introducing additional safety concerns or risk to the patients who have already suffered from this infection disease. So putting together all this information, we believe it is safe to conduct based on the safety profile. That's one of the major reasons we decided to move forward. And in the same time, we actually have also obtained China regulatory's approval for conducting phase 1B trials in the rheumatoid arthritis patients with the similar dose ranges. So that's why we believe the dose we can use for the patients should be safe and well tolerated as well. One of the other reasons we can quickly move from China to US is because our company have capacities conducting clinical trials both in US and China. We have a team located in Maryland, Rockville, where they have a full capacity of running clinical trials. And currently we have 2 active studies executed by those teams. So with both teams support both US and China, we believe we can have a quick turnaround of global trials, either in US or in China. So overall, we have about more than 100 staff exclusively in the clinical development with all the functions needed to support the regulatory interactions, to support the discovery and the translational medicines, and then to support the clinical protocol development and executions. So this particular GM-CSF trial for supporting the treatment of COVID-19 started in China. However, because of the quick control of this outbreak in China, we decided it make more sense to move to US where the pandemic is just started. So that's why after we had all the packages ready, and then we quickly moved to US for consultation. And then we had very quick interactions with the regulators and with the PIs and the frontline physicians. We quickly wrap up with a meaningful package for submission. Here is how it's our strategy and the whole route, how we come to where we are now. So the first is the study I mentioned earlier, is the US phase 1 study. We have healthy volunteers in the dose cohort escalations, which completed last year. As you can tell, we have a very acceptable safety profile, and our PKs increase linearly in the proportion, which is very ideal for further development. With that data, we come to China, propose another studies, which is also approved for rheumatoid arthritis. But this is just keep in mind, RAs is not just a we wanted to develop solely, and we believe RA is just a proof of concept. We already planned to do first like cytokine release syndrome. At the time was for the CAR-T related, but now we are expanding to the right side, the 3 global studies. First is we started in China because the outbreak control, and we decided to move to Korea and the US. So right now, the Korea is reviewing our submissions, and then we hopefully will get the response quickly. And for US, we submitted in the middle of March, when we received, actually no, at early March, but we received the approval very quickly afterwards. So if you calculate the working days, it only took us 3 days to get FDA approval. So it's very impressive. We truly appreciate the officers in FDA agent worked very diligently under these circumstances to help approve or moving this project forward for the benefit of the patients. You may ask, what is the study design? And we had gone through a lot of thinking. At the beginning, we thought about passionate use, which would be the open label study, just like what Italian studies did. And then, however, we believe, to answer scientific questions, it really requires a randomized study. So we then set up the study design with 2 stages. First stage is the open label, and the second stage is the randomized study. We went to the pre-IND discussion with the FDA, and then their feedback is they suggest that the first and second part both are better off with randomizations, because otherwise, if there's side effects or safety concerns, how would you differentiate it from the disease? So we took their suggestions and then decided to set up this two-stage study, but all randomized, double-blinded. So we called here placebo is actually like standard care of those patients. We don't want to introduce too much complicated protocols to interfere with patient treatment. So we let the physician decide the standard care, and then on top of that, we introduce two other arms. One is 3 milligrams, and another one is 6 milligrams per kilo. One single dose shot for those patients. Keep in mind, as I introduced earlier, we have finished our first phase 1 studies. The dose range is 1 to 10 milligrams. So we believe this 3 or 6 milligrams per kilo is very safe for the patients. So we had these two parts. First part is each arm with only 8 subjects. After we're completing that, we have an independent safety committee to do assessment and decide how to move forward. And then the second part will open up for more patients. As you can tell, it will be over 100 patients to give us solid signals if this the compound work and if it's safe. So from there, we will discuss with regulatory agents to see how to move forward. Considering the urgency of these situations, we believe it is much more flexible in terms of when and how to communicate with our regulatory agents, especially those reviewers who have been very willing to discuss with us at any time. So this is the current path for the studies we had initiated with the sites. We have also obtained the regulatory and ethics committee approval in US. So it's fully initiated, and we will start the study at any moment. So in the same time, I just wanted to share recent progress in China. Because China, even though the outbreak was under well control, and then the government and the country believe that we need to prepare for the next outbreak if it happened with any chance. So we have been invited to join the studies for a potential utilization just in case it is needed in the future. So we are thinking to invite make China as part of the global studies together with US. So in that case, we can have a more meaningful data generated from different populations, and also more efficiently to come up with the patient number needed. So in summary, I think these projects just gives us a very good lesson. I believe I have never encountered like that. So we initiated in the very early of February, and in middle of March, we were able to initiate the study in US. So I think it just tells the urgency of this disease and how how difficult it is for dealing with this unknown disease, because we don't have a clear guidance before. We don't have approved drug, particularly for it, and we have to act very quickly, whatever we can, to find the niche or the potential to treat the patient. So it just I'm very pleased that we are just joined part of the world to be part of this fighting against this illness. And then we don't know if it works, but we do it with the most scientific way. First, to ensure the safety of the patient, and then secondly, to design a study scientifically sound, but also practical. And then thirdly, to utilize the capacity both in US and China to set up this strategy and studies to go wherever most suitable. But ultimately, I think it's for the benefit of the patient in the world. So coming back to GM-CSF antibody, we believe it has a great potential to treat CRS, cytokine release syndrome, regardless of what is the cause of this illness. But the presentation are similar, which is cytokine release resulted the chain of events in the like inflammations because of the immunodysregulations. So, however, at this moment, we do need to understand the disease better, and we will continue to collect the information, hoping that the data itself will speak for the effectiveness and the safety profile, and hoping to find a solution or options for treating the patients who are needed the most. And I think I will stop here, taking any questions if you have. Oh, the first question is they ask what is the quantity of this antibodies currently we're producing if the needs are increased after the coronavirus infection outbreak. How do we increase the productions or manufacturings and to satisfy the clinical needs? And I will try to answer is, right now we have enough to support the entire studies in both US and China. And then, like I mentioned, because the original study was in US, we still have quite enough to support the study. And then we also are manufacturing another like a batch, which should be also at least about another like 1,000 batches, which should be sufficient to support the immediate needs of the public if it's efficient, as we had discussed. The good thing was because it's manufactured in China, and then we have been closely working with the CDMO for this. So we believe we have much better handle or control in terms of the manufacturing and its quality, and as needed. We have been told by CDMO and our own CMC team, we can prepare additional manufacturing as long as we were told like 6 months ahead of time. So we are very comfortable with this. And then the second question is asked when the antibody can be approved or commercialized, and if there's any way to accelerate it. Well, this one I can only speculate with you because the current study is designed as a phase 2. And however, I believe, based on my knowledge with interaction with the FDA, if it goes well, if the data come out as we have predicted, we can negotiate with the agent if the pandemic is still there and the patients are needed. And we can probably have conditional approval, for example, to get to the patient public, or we can expand it to the global studies when other countries can join. So then the data will be more solid. So actually, we have some of the KOLs in China already discussed with us, said, well, the government wanted something available in September. I think it's very aggressive this September, but if the data comes well, and then the Chinese like government or regulatory agents seems to me are very willing to discuss the green channel to provide it to the patients when it's needed. So I would not give a definitive answer, but I definitely believe there is a potential green channels for either in US or China or like Korea, other countries. So based on the willingness of their conversations with us. So I'm again very positive. And in terms of what is a good option to choose? Do you think HCQ? I'm sorry, I'm not very familiar with HCQ. What is that? So the question is, do you think HCQ is a good option to treat? Oh, you mean, oh, I see what you mean. Hydrochloride quinine. I think there's a certain value for it, but I think you have to select patients very carefully because of the potential side effect. Especially if the patient had some basic medical conditions or concerns, like prone to these side effects, I would be very cautious. But otherwise, if the patients are relatively healthy, can tolerate those side effects and without any other efficacious treatment, I would try. Otherwise, I think it's just a risk and benefit assessment. See how you feel comfortable. I think this is always a dilemma for physicians to treat patients. When you don't have any options, anything can help patient, you will try. I think that's where the position I would take. And so other question is, what is the antibody difference, like special, do you have any like differentiation on your antibody design in terms of this GM-CSF? I have to, but right now we believe this is the first antibodies coming from our own antibody platform. We believe it's one of those best in class because the safety profile is better. That's why actually we designed this compound to begin with. But as to the special on the antibodies, I would not be able to share with you today. And next time, if you have interest, then we can discuss it with our discovery folks who can tell you more. I don't want to mislead. But to me, this antibody definitely, I can share with you with the phase 1 data from US. Besides the safety profile, the PK and PD is very pretty. I think the CSR is just recently had completed. We should be able to find the venue to publish or present. The PK/PD profile is very good. So that gives us a big safety window to conduct other studies, and under the notion that it's you can just play around among a larger safety windows for this compound. So let me see what. Let me go down. Okay. This is a. 请问您3年 SARS 的时候用过抗体 SARS 引起的 CRS 风暴日有当时结果会什么样 ？Okay, this question is asking if the SARS anybody has any experiences or data showing the CRS triggered by the SARS. I don't think so. At least I didn't see any data coming out of that. So this one is only recent years we knew from the CAR-T treatment that we had a better understanding of this CRS. So at the SARS, that was over 10 years ago. I think, we learned everything from this time. That is a good question. What will be the price? So actually, we did that first, I would say, is under the, our own aspiration to find a solution to treat the patients. It's our social responsibility to do that. Actually, the company pulled the budget out of others to prioritize this. Because we originally did not plan this compound and the studies at all. And actually, originally we only planned in China, later on become like global studies. So it is expanding, but we are proud of ourselves and able to contribute to this, war against it, against the virus. So at this moment, we are not making any assumptions of the price. So we'll go to make it public available first. So even we had some preliminary discussion with some of the Chinese officials. They said if we develop, we have to make it public available and without negotiating the price. We already said yes, we would do that. So at this moment, I think sounding the commercial success is much less important than making it available to the public. Production rate and cost. So the next question is from production rate and cost. Are there plans to carry out more trials in Europe? I cannot tell you such a detail about this production and cost, but I can tell you because it's manufactured in China relatively, it's manageable in terms of the cost, and the yield is pretty good. And then, so the question next is, do you have any plan to carry out more trials in Europe? Yes, we did. We actually, it could be potentially more countries. We thought about Italy to begin with, but then we had a preliminary consultation. It looks like they have, you have gone through a lot of preliminary assessment by ethics committees, and so because it's such a novel compound, and by the time if we got the turnaround consultation results, the situation could have been come down like China as quickly. So we decided to do that in China, in US first, and then decided where it needed. So, but I think to us, to running a global trial is not a problem. It's just a matter of how to best move forward. And next question, any difference in terms of regulatory requirements for FDA and NMPA? Well, what I can tell is both of them are acting very, very quickly, and then they both created green channels to facilitate the process. I think NMPA definitely takes more cautious steps in terms of assessing this brand new compound, and then which just newly got the IND approval in China. So they suggested us to do more preclinical data, to run more preclinical studies before they feel comfortable to introduce to humans. And when we went to the US, the regulatory reviewers at the beginning, they also said, wow, it's very new. Why don't we? They strongly encourage us to do pre-IND consultation instead of submitting to avoid any delay. And then we wrote the questions. The first question is, do you think we need any preclinical studies to support this IND? Their answer is very clear, no. So that means we don't need to do any preliminary or preclinical studies to support these clinical studies. I think these are the major differences by these two regulatory bodies. So I think it's routine, normal in China NMPA always asks for conducting adequate preclinical data in terms of its tox studies, in terms of its efficacy, animal models, or any preliminary in vitro, in vivo studies. So, but because this is, COVID-19, we don't have any animal models. We don't, have any, time to run these preliminary studies. So that's just why we ran out of time in China. So, but I think both, we are very impressed that both of those like agents worked very diligently to help people. Yeah, I got the hydrochloroquine. Yeah, the question is already answered, like I said. Next question. So next question is they asked if the on the cytokine release syndromes, how do you know GM-CSF is the most important? And in a during the inflammatory response, each of them says it's more important. But well, in my understanding is, first, the GM-CSF stands on higher hierarchy, which you can look at this if you're interested in this biology and immunology. It does stand on the higher hierarchies. The graph I showed you guys earlier is not made by me. It's by other scientists in China. So it is first, it's a higher hierarchy, just like a switch on a higher level. And secondly, I believe every patients are different. So GM-CSF, since it's on a higher hierarchy, it has more chance to affect this, severeness of this, the cascade of cytokine release. Not saying IL-6 is not important. Maybe, based on all the collective data, some patients had, almost all the patients had a GM-CSF increase, in a larger amount. But for IL-6, some studies showed IL-6 increased, some other studies showed IL-6 reduced. So again, patient profiles are different. Like even the cytokine release syndrome could be, some patient had more on the, GM-CSF release, some others had more on the other, IL-6, IL-2. So anyway, not consistent. So, but overall, the chance of having, GM-CSF antibody treatment could be larger. But we ultimately, in the end, I think his data itself will speak for it. So in our study, we will definitely measure the GM-CSF and measure IL-6 and a few others to see, what is the change through the course of the treatment, because that will be a very important biomarker to show, definitely if the treatment had resulted in the reductions of the cytokines. So in addition to the clinical improvement. So I agree with you. There's no definitive answers, but in the end, we'll see the correlation with the data. So let's see. The next question is, that's a very good question. It says treating the cytokine release really requires the good timing and then good, like dosing. So how your company control this? Yes, we had a lot of discussion about, what dose to use and how to measure when to introduce. So in terms of dose, we had struggled between should we introduce 3 milligrams, 6 milligrams, or 10 milligrams, or should we use like if we have given one dose and then space off second dose and third dose. So actually, it will be ideal, if you didn't give one dose, the patient does not respond and give second dose. But right now, we don't have a very good, protocols to use to have the physicians use very appropriately, like when to introduce a second dose, because every patient is so different. And then we have limited understanding of the safety profile of this compound. We don't want to have a multiple dose which could be potentially accumulated, causing more toxicities or side effects and then benefit. So we discussed with our regulators and physicians, and they agreed. That's why we have two dose cohort. One is 3 milligrams, and the other one is 6 milligrams, just single dose. Because of the half-life of our compound is very long, so we decided one dose should work if it's on an adequate dose range. And in terms of the window time, it is also very critical. So because this is phase 2, we wanted to make it as simple as possible, but we also don't want have a patient who were given this compound not unnecessarily. So it's a very, very hard decision how to, when to introduce it. So then today I didn't have enough time to discuss with you about the multiple. We use the compensatory measures to decide if the patient is eligible. So we, for patient from I think one of the major measure is the oxygen saturations. So once the patient started to have oxygen saturations less than 93%, for example, and then we believe the patient requires like exploratory support. In that case, we should start it to intervene with this compound to prevent it from further deterioration. So there's a clinically it's very hard, but the good thing is this is phase 2, and if we believe then the patient can be beneficial, and early intervention is better than later, and then we can either use interim analysis or in another phase 3 to adjust to the better window time. So that is one of those challenges clinical, because again, when you are having the patient who needs the adequate treatment, you don't want to give too much protocols to make it more complicated for patients. So then, and in the same time, also making sure patient doesn't have to expose to unnecessary treatment. So this is kind of balance we are always facing. So another question is, how is the safety profile of your antibody better than the others when some of them have hundreds of patients through in the phase 2 until in phase 1B? We currently are in the phase 1B and 2. Yes, but that's a very good question. But keep that in mind. Our current study is also single dose. So even it is not traditional phase 1B anymore. So it's a therapeutic dose. It's a therapeutic, regimen. So if, for example, if we only in phase 1 now moving to phase 2, we need to have multiple dose. We have to having a dose escalation. No, we don't need that anymore. So the single dose demonstrated safety profile in our phase 1 study is the same dose, actually much lower. In the phase 1, we had gone up to 10 milligram per kilo. Now we are doing 3 and 6 milligrams per kilo. So that is a much safer dose range. We don't give a second dose. So that's why we believe in general it's safe, but we worried actually more about efficacy. So, but none of the GM-CSF has been in the phase 2. So we are all in the same, for this indication. I agree. Other GM-CSF for RA indications having more safe phase 3 safety profile. In that case, definitely that is better safety profile. However, it's a different patient population. So we will need to continue to explore in these patient populations. But again, I want to emphasize our study is a single dose and much lower than the ones we have done in the phase 1. So the safety profile, I have more confidence. Are there any approved GM-CSF antibody out there already? No, none of those. So all I know is recently GSK had completed phase 3 studies for RA, but none of those are for this COVID-19. This question is, FDA have any fast track for this approval? I think so. If you see, we've got 3 days for being cleared. 30 days is an average for the standard for IND clearance. But this time they particularly gave us 3 days, 3 working days. I remember, we had submitted on a Friday in the evening. So that means if you don't count the weekend, we got approved on Wednesday. So 3 days. That definitely showed that they wanted to support the drugs which can be potentially treating this disease. And then they had also opened dialogue, says anytime we had any good results or information, we should always go back to discuss with them. So I'm pretty positive if it works, and then we can have a fast track. Humanigens, as far as I know, they also initiated Nizumab. They also initiated a study for treating cytokine release syndrome in COVID-19 recently. I think, if I remember right, they had a news announcement. So almost all of those have the GM-CSF have joined the clinical trials for these indications. The Nizumab actually in their previous CAR-T related cytokine, they used it to both use prophylactic treatment and the actual treatment, because they believe if you block, just like switch the GM-CSF earlier off, then you can prevent from the cytokine release. So we are hoping also in the future can move forward, like I mentioned earlier, to the earlier phase that, to help patients even before they become severe. And the next question, will block both L6 and GM-CSF at the same combo be more effective? That's a good thinking. In the future, it could be, but at this moment, as I mentioned earlier, because some patients showed IL-6 increase, some of others don't. And then as we have more knowledge of how the cytokine release has been, the case in COVID-19, we should be able to better understand if this combo will be a way to go. And that's a very good thought. And we actually were thinking of that too. But, now we want to sort things out first. Can we inhibit JAK kinase? You mean GM-CSF? No, we are very laser focused. Actually, we talked to some of the professors in China who have led this areas development, and in his conversation with me, he said, well, yeah, people have recommended JAK inhibitors, have recommended L6, but I only recommend your company's GM-CSF because you are more focused on the cytokine release without affecting other normal cell functions. So in that case, we truly can limit the side effect or suppressing other immunofunctions. And he believed JAK, kinase inhibitors expressed more profoundly of those immunosystems. So that should not be the ideal situation to use. Even though, the currently JAK inhibitors have also been approved or not allowed to do the clinical trials in this situation. Did you address the neutralizing antibody issue yet? We have, you mean the ADA? No, we have not observed anything yet. But I mean, in our phase 1 data, we have not seen the ADA issues, if that's the question you're asking for. We'll continue to collect the data, as more longer patient treatment coming along. So right now, so far we haven't received received any information from our phase 1 data. So you are saying l-Mab is in phase 3. I think I thought they just initiated study, but I didn't realize it was phase 3. So the only thing I can think of is they used the cytokine release syndrome data from the CAR-T to bridge that into the COVID, like a phase 3. Otherwise, it couldn't be as quick. Yeah, I did see the announcement, but I didn't realize it's phase 3. I apologize. I'll have to go back and study. Make a bispecific against both. That's a good idea. But the problem was we actually had some bispecific platform. We had a real struggle to define the dose of these two bispecific, especially cytokine, because when you come to the bispecific, the dose is very critical. And one of those bispecific it come out is the dose, the toxicity becomes much larger, bigger, when it comes to bispecific, when it's separated, two compound is very benign. So it's not impossible, but it requires a lot of work. You have to mutate this cytokine to make it less toxic or to match these two. So, and also every patient is different. It might be hard to use unified the bispecifics to treat all the patients. Yeah, it's a good thought, but as I think the technology become more mature, we can manipulate this more. So what is the half-life of this antibody? The antibody is very lengthy. I think it's as far as I know, it's about 20 some days. So that's why one of the reasons one injections can last very long. So our antibody is particularly long, much longer than the IL-6, as far as we know. Yes, this antibody, we actually, can this antibody be used for CRS caused by cell therapy? Definitely. That one was already under discussion with one of those partners, because our company, we don't have a cell therapy, but we were in discussion with another company who had these assets, and they are interested in co-develop this compound for these indications. Definitely. That's how actually the original ideas came out for the COVID-19, because we saw the similarities of the CRS in these two diseases. And actually, if you go back to the humanized, I think they also, because have done, because ahead of us in terms of cell therapy, they already accumulated some information about this, especially safety profile. That's why they can initiate studies faster. But we'll see. I think we ran out of time. Yes, again, thank you very much for your time to discuss. And I can tell we have a lot of questions. Unfortunately, I cannot answer all of you, but it's a good exchange of information, and hoping we all contribute to this particular like a special war against this virus, and hoping we find solutions to help the people who are needed the most. Okay, I will start from here, and each of you have a very good day in the East or US, or a good night in China. Thank you.